Hi, everyone. So now we're at this fortunate place where we have all our speakers except Jane, I believe, to discuss the questions we didn't get to and have an overall general discussion from the topics that have been covered. We've been really fortunate to have 6 different countries represented in this session alone. And there's certainly so many different ideas and research that have been raised. So maybe we'll start with some of the questions that we didn't get a chance to discuss during the session. Woong-Yang, do you have a suggestion on where to start with that?
Okay, I got the question from Jay for my talk, BA. He asked about the some maybe cell composition between responder and responder, the difference in the baseline. Then maybe you can use it as a predictive marker. So yes, and then we can use that for the predictive marker for the immunotherapy or chemotherapy. But the limitation will be we need to use that a single cell technique to use that signature from our findings. So maybe we need to translate that. For example, we need to develop the panel or other method to catch those signals for predict some response to those the therapies. So it's a good, the purpose of the study BA, we want to develop the method to predict that patient outcome with the the markers. And I think maybe can ask to Jane from Christine. Actually, she asked that Hoib and Lubeck have multiple layers of regulation. How might this impact on its suitability as a therapeutic target? Yeah, can you answer to this question, Jane? Yeah.
I think Jane is not with us. She wasn't able to make the session, but instead, perhaps I can ask Professor Kataoka a question that was put up here. How is the PD-L1 protein transferred from tumor to surrounding cells? Does this process happen in vivo?
Yeah, so thank you for that question. So we think PD-L1 is transferred through both direct contact and indirect mechanisms. And the latter mechanisms include, as PD-L1 is known to be secreted in exosome. So the latter mechanism, so indirect mechanisms include those mediated by exosome. And in addition to our human single cell data, so we have created a genetically engineered mouse model harboring PD-L1 3' UTR truncation, so the same genetic alteration expressed only in CD4 T cells, and confirmed the messenger RNA independent surface protein upregulation of PD-L1 in that model. So we confirmed the process also occurs in vivo.
Thank you.
And maybe question to Murim, BA from Jane, that what are possible confounding factors that contribute to the NAFLD-specific regulation of AGXT2, but not in health control?
Yes, that's a good question. We were very interested in understanding what would be the upstream regulator that only turns on the EQTR only in the NAFLD tissues compared to the control tissues. So we tried various different experiments, and we trying to look at the differential expression of isoform of AGXT2. And also we trying to look for the transcription factor binding of the SNPs. And also we were epigenetic modifier and so on. We also even put those SNPs in front of a luciferase reporter and trying to see whether these SNPs change the reporter strength in the NAFLD model. But sadly, we didn't find a very definite answer yet. So we don't really know, maybe a little bit of everything, maybe, or not. So another issue is that because the SNP that I showed you is in complete LD with other neighboring 5 different SNPs. So it's really hard to pinpoint what SNP is exerting the biological function.
Okay.
So perhaps I can ask the next question for Ying. So that was a really brilliant talk. And Shams asked, he was intrigued by the low ARI of the Surat pipeline. There's been a few published papers that claim to cluster these well-known benchmark data sets almost perfectly. Have you tried comparing to those?
Yeah, thanks a lot for the question. So actually, for the pancreatic data sets from the MNN paper, so actually we have observed, so the latest version of Surat actually performs better than the early version of Surat, but nevertheless, compared to the FACS labeling provided by the original publications, so the AI is still not that satisfying by Surat. So the recent, the Surat pipelines that we recently looked into, so they actually ask the input of the sample conditions for the cells. But nevertheless, we have noticed that actually in the batch effects correction step and the clustering step, they actually don't use those information. They only use those sample conditions in the calling of differentially expressed genes. So from our perspective, I think it will be better to consider those sample conditions in both the batch effects correction and the clustering steps, so that the accuracy can be better. So in our simulation studies, we also observed Surat does not perform that well. But of course, you can say a simulation study can favor our setting. So we will, of course, look into more benchmark data sets. Thank you very much.
Great. Maybe it's time for the discussion for non-infectious disease and then in HCA Asia. So maybe we can think about some potential to collaborate together in Asian countries to for the facilitate the research and then development based on the single cell techniques. So as we discussed yesterday about IDA, so we experience a lot of some Asian specific or some ethnic specific, some difference between countries, even in Asia. So I think maybe another issue would be cancer. So I think I would raise the issue related to how can we work together for the cancer together.
I think if I can add, I mean, I think one of the strengths of the Human Silenced Asia community is this ability to really focus on what helps our regional area, you know, in terms of the populations that are represented. And it was really nice to see across these different talks, the focus on cancers that are particularly relevant or important to Asian populations. And so, you know, in thinking further, are there diseases or other conditions or environmental exposures that are missing in that list and need to be a focus for our HCA Asia community? And Shams just popped a question there in terms of, is anyone doing single cell analysis of CML? There's a keen interest there to collaborate.
Yeah, these are the approach to raise the some type of cancer. We can work together because most of the cancer in Asia be not prevalent. So I think maybe we can work together to combine the data or maybe collect the sample together, right?
Yeah. So can I ask all our speakers just to stimulate some discussion? I mean, how did you arrive on the cancer type that you're working on? Like, Woong-Yang, how did you choose to focus on gastrointestinal cancer? Was that because of availability of data or collaborations that you already had, or like what gave rise to that? I'm wondering if each of the speakers can talk on that topic.
Yeah, actually, gastrointestinal cancer is a Korean. I mean, highly prevalent in Korea. So especially the gastric cancer be one of the most common disease. Also, yeah. So maybe we get interested about those things. So, but also it's not common in other, Caucasian, Western countries. So it's hard to get into the top, but actually maybe it's kind of some our strength to apply the technique for those the rare disease in the yeah. So,
absolutely. And Sumitava, was that the same with you? You mentioned that the cancer that you're working on is relevant to the Indian population.
as you know, so still we do not know that why. I mean, there's some environmental effect for the some Asian specific cancers, but still do not know the some genetic or some effect on our environment together. So maybe probably that Ignatius actually he collected data from many countries based on collaboration, but actually you experienced the kind of how to collect the data and how to combine the data, right?
Yes,
So,
and share the data too, right?
Yeah.
Yeah.
Can you share your experience with that?
Well, actually, it's a quite interesting experience because we are working from people from 3 different countries, and then we have like 5 different cohorts, and then yeah, there's also some challenges in the sense that there is multiple huge batch effect because of this different countries and a different pipelines. Sometimes they were trying to analyze the data as well, not analyze the data, but to collect the sample and then process the sample, right? But yeah, it's been very interesting experience for me because we have a very diverse people with a very diverse background as well, and then yeah, hopefully we can keep doing this, and then the progress that we can do with this kind of collaboration is also massive in the sense that we are up to date to the thing in the field, and then we have more support when we have some problems. So I think it's very beneficial overall. Thank you.
I'm curious then, Yiging, I'm not sure if you were listening to the talks yesterday, but Sham introduced the ADA project, which is looking at understanding immunity in different Asian populations. And he was saying how important it is to find ways to reliably address batch correction effects. Because if we want to understand immunity and use the different countries and all the data that we have, we want to be able to address the batch effects there and really leverage this meta-analysis. What are your thoughts on these big studies that cross different countries that have all these other things? Do you think it's possible? Are you hopeful that we can come to a place where bioinformatically we can reliably use the data?
Sure, I think that's actually I think that's also the fascinating part, right, for the data integration. So I think in general, when we do meta-analysis, usually we want to pull the information, pull the samples, right, to improve our power for detection. But meanwhile, sometimes we also want to keep the study-specific signals. So I think when we have those large consortiums across countries, so I think one interesting thing we can investigate is whether there will be any country or region-specific signals. And meanwhile, so we also want to prep for the batch effects to pull all the shared signals. So I think the large cohorts consortium efforts will be really beneficial. So then we can see what will be the common disease mechanism and what will be the country-specific and the population-specific signals. Yeah.
That's really, really great to hear. Do you think it's something we can achieve with the current kinds of methods, or do you think there still needs to be more bioinformatic innovations? And we have about 10 seconds, so really quick thought on that.
Yeah, I think nowadays, right, so on bioRxiv, you can see every day there are tons of papers comes out. So I think different paper based on different assumptions. So I think we also need to validate all those assumptions more carefully in real data sets and see what assumptions fit better. Yeah.
Yeah, absolutely. Awesome. All right. Well, I'm sorry to cut this discussion short. I feel like we've only just gotten going. But I'd like to thank all the speakers in this session. Woong-Yang and I are really pleased with how the session went. And thank you so much for your discussion. And we look forward to resuming after the break.
Okay. Thank you.
Thank you.
Thank you, Bob.